मैं उस पे चल रहा है ना. Good afternoon, students. Now we are starting with the first unit of the industrial biotechnology paper. So I hope many of you would like to opt for this paper as an elective subject. And we are taking up the first unit, that is isolation, screening, and maintenance of industrially important microbes. So this is the first lecture of the series that we are going to take up in this first unit. Now, what is isolation or screening or maintenance? Why do we need to know that? Now, isolation, as such, we are having a diverse variety of organisms that are existing in the nature, and each organism is having a different characteristic. Some of these organisms are of importance for the public for commercial use. For they can be used for getting their secretory products or any kind of products that can be helpful for the mankind to better their uh, development. So, but how do we identify those species? How will we get those species that are really beneficial to the human beings? Not all of the species are beneficial. So first we need to identify the beneficial species. Now, how will we identify those species? So the first and the foremost thing is that we need to isolate them. The isolation is separating in industrial terms, if we talk of it, then it is the separation or selection of the species that will be commercially viable, that will produce some kind of commercial product for the use of the mankind or the other species. So we are trying to obtain those particular species. Now, the result of isolation is that either we are getting a pure or a mixed culture. Pure or a mixed culture means pure is species of a particular uh, genus. So that is a pure species, a pure strain we call it in biological terms. Or in a mixed culture, there are variety of organisms that are being found in the mixed cultures. So the mixed culture is not a single strain, it is rather a combination or a mixture of various kind of strains. So that isolation results in obtaining either a pure or a mixed culture. Then this isolation, when you isolate the cultures, what you are going to do with that? If you have isolated a species, you need to use it somewhere, you need to apply it somewhere, then only the isolation process is required, otherwise you don't need it. So when you have isolated the species, then you have to particularly assess its feasibility also, whether you have isolated the correct species or not. Correct in the sense that the cultures will carry out the desired reactions, or they will produce the desired product. Desired reaction, if you are hoping to carry out a lactic acid fermentation, then that particular organism should carry out the lactic acid fermentation reaction. If it has to produce an insulin enzyme hormone, then that particular organism should produce the insulin hormone. So you need to assess whether you have received or selected or isolated the strain that will give out the desired reaction or the desired product. Then, when you talk of things in the industrial terms, then the basic thing is that we are talking of is money or finance or economy. So, the isolates, that organism that you have isolated, must carry out the process economically. That is, whatever inputs you are putting in for the process to continue, and then as a result, whatever outputs are you are getting, that if you take the difference, then there should be some profit for the person who is using these techniques. So if there is no profit, that means doing all this process is waste. So economically, it should be feasible. The isolate that you are isolating should carry out the process economically. That is, its media requirements, its strategic requirements like temperature, humidity, pH, or other nutrients. That all should be very low cost. Then finally, when the product is being produced, then that can carry then the purification processes or extraction processes, that should also be economically feasible. So in all, when you total up all the things, then the process should become a totally profitable process. Only then it will be known as economical process, and the variety or the strain or the organism that will do this will be your best isolate to be used. Next, now you want to isolate an organism. What should you think before you are trying to isolate the organism? What should be your criteria for the choice of the organism? First is the nutritional requirements of the organism. Nutritional requirements, whether whatever substances or substrates you have available throughout the year in your immediate environment, whether that organism will survive on those immediate resources or not. If you have ample stores of glucose rich substrates and the organism can survive on those substrates, then it is very good. If not, suppose it will survive more on sucrose rich substrates, then it won't be beneficial because you have to export or bring in the product from some distant place that would be rich in sucrose, and then the cost of transportation will be added. So you cannot take up that organism which does not survive on the locally available resources. So the nutritional requirements, then it should not have, suppose this was a simple example of a glucose or sucrose. Now if there are some specific requirements, some specific hormone or a growth hormone or some kind of vitamin or some kind of heavy metal that is specifically that organism requires, then the cost will be added. So the organism should require a simple nutritional substrate. The stringent requirements for oxygen for particular kind of gaseous environment should not be that rigid. Only then the organism will be good organism for your processes. Then the temperature range of the process organism. Now, the process organism should uh, particularly work at the optimum temperature should be uh, around the place where you are trying to carry out the process. Suppose the ambient temperature is around 27 degrees centigrade and the organism is working at 37 degrees centigrade. That means you have to provide extra heat, or you have to increase the temperature so that the organism can work to the optimum level. You have to increase it from 27 to 37 degrees centigrade. That 10 degree rise in the temperature means you have to put in efforts to heat up the whole environment or the unit where the process will be carried out. So the temperature range should be somewhat near about the ambient temperature. Then the organism should be able to survive in particular ranges. Suppose the process, when you start the process and when you end up with the process, there is a temperature difference of 20 to 25 degrees. Then At 25 degrees, particularly, the organism will be killed. You are starting from 27 degrees centigrade, rising it to 37 degrees centigrade so that the organism work at the optimum temperature. But when the temperature rises from 20 degrees to 25 degrees, that means the optimum will reach from 47 to 50 degrees somewhere, and then the organism won't survive beyond 37 degrees. So at that particular temperature the organism will be killed. So you have to have an organism that can tolerate at least 10 plus minus 10 degrees, 5 degrees temperature, so that you don't have to strictly maintain the temperature requirements at each and every step of the process. So that is the importance of the temperature. The optimum temperature of the process organism should be uh, taken into consideration. Then the suitability of the microbe to the process and the process equipment. Now, suppose you are trying the microbe, you are using the microbe for production of some kind of alcoholic beverage. Now, if this microbe is susceptible to the higher concentrations of the alcohol, then the microbe will be definitely killed as the alcohol concentration rises. That means the microbe will be killed at that particular step or that particular instance. The process will be halted. So, the microbe that is producing either an alcoholic concentration or an acidic environment, or whatever in kind of particular environment it will be producing as a result of the production process, the microbe should survive that particular environment. So, it should be suitable for the process. Then the process equipment. The process equipment that we are strictly concerned with is the fermentation vessel or the container in which the process will be taken up. So the process equipment, whether it is of metallic or glass or fiber or whatever it is, the microbe should survive in that compartment or container. It should not be that when it produces some acidic product, the leaching occurs or the rusting occurs in the vessel, and as a result of that leaching, the microbe is dying off. Some toxic substances are being delivered in the media contents, and the microbe does not take up that toxic substances and it will be killed. So, whatever equipment you are going to use, whatever material you are going to use for that designing that equipment, the microbe should survive in that vessel. So, it should be suitable for the processes. Suppose you have the availability of a glass chamber, the microbe should be able to survive in that glass chamber. It should not be susceptible to the silica in the glass chamber. Then next is the genetic stability and the amenability to genetic manipulation. Now, if the strain that you are isolating is highly mutable, that means we are today this strain is particularly producing high contents of acid. When it mutates, it loses its or the capacity or the ability to produce that high content of acid. Suppose oxalic acid we are talking of. So if that ability is reduced, then the microbe is of no use. So the DNA content or the genetic component of the microbe should be highly stable. Second thing, that when we talk of genetic manipulations, amenability to the genetic manipulations, that is, you're trying to insert some kind of foreign gene or you're trying to change the genetic composition of the microbe. So it should be available for that genetic manipulation. Stability is different thing. That is, it is not prone to the environmental changes or the changes that are occurring within the process system. And amenability to genetic manipulation is, that is, when you are specifically trying to design that particular organism for a specific purpose, then it is available for that modification. It can easily be modified for that purpose. So that ability should also be present in the organism. Then the productivity of the process organism. Now you have given ample of substrate options, you have given ample of process criteria, different temperature ranges, pH ranges, or salt concentrations, and so on and so forth. But still the productivity or the efficiency level of the process organism is not too much. That means this process organism won't be liable or available for the economical utilization if it is not efficiently utilizing all the resources. So the productivity of the process organism should also be very good. It should utilize the maximum substrate, the maximum available resources to produce the product rather than the side products or waste material or excretory products. Whatever your intention is, if you are producing the citric acid, if you're trying to produce vitamin B, then it should specifically produce that particular product in large quantities out of the max of the substrates available, not rather some other byproduct is being produced in higher quantities. So that is the efficiency of the process organism should also be very high. Then the ease of the product recovery. Now when you are trying to isolate the product from the at the end of the process, then the organism should not create any kind of hindrance. Suppose hindrance where, now if you have the microbial size, if you consider and you consider the size of the product in case it is a solid product, then maybe you are trying to use the method of recovery as a filtration. The method of recovery is filtration. Now if both are of similar size, then you won't be able to filter out the product or the microbe or separate them at all. So it will interfere with the product recovery. So the organism should not interfere with any kind of downstream processes when the product is being formed and extracted for the commercialized use. So these are particular six few criteria that you need to consider when you're trying to choose an organism for any economical or industrial process. Now we talk of the isolation methods. Now you are trying to isolate, now you're trying to isolate the organisms. Now how will you isolate these organisms? Firstly, you are trying to isolate these organisms based on the desired characteristics. Suppose you want to produce you want some organism to produce antibiotics, then you need to select that organisms that will produce that particular antibiotic. Now these now all these organisms that will produce a particular antibiotic, suppose penicillin you are talking of, if all the microbes that you are isolating are producing penicillin, then you can take them as isolates. Now all these organisms that will produce penicillin will be the organisms of your choice, so you will select them on the basis of the characteristic, that is the production of penicillin. Second is on the basis of not selecting desired characteristics. Now, if you consider all the isolates that are producing penicillin, all of them won't be beneficial for producing penicillin on economical purposes. So, for that purpose, now production of penicillin cannot work as a criteria for isolation. In that case, you need to select the organisms uh, or that won't produce penicillin, or some other kind of characteristic you need to consider so that the desired characteristic is not obtained. You won't take the organisms that are producing penicillin specifically. Rather, you will now take them on the basis of the substrate selection or some kind of other criteria, on the basis of which you will select those or isolate those species. Then, now first we take up the isolation on the basis of selecting desired characteristics. Now this is particularly done with the help of the enrichment cultures. Now the enrichment cultures is that you are providing some kind of additional input to the growth medium so that the particular species only grows. You will provide some specific substrate that will favor the growth of a specific species or a specific strain. Now, this is known as your enrichment culture. Mostly, you try to carry out these enrichment cultures in the shake flask. Now, this will result in the growth of the desired type from a mixed inoculum and result in the modification of the medium and therefore change the selective forces which may allow the growth of other organisms is still viable from the initial inoculum, resulting in a succession. Okay, so now the selective forces may be re-established by inoculating the enriched cultures into identical fresh medium. Such subculturing may be repeated several times before the dominant organism is isolated by spreading a small inoculum of the enriched culture onto solid medium. The time of subculture in an enrichment process is critical and should correspond to the point at which the desired organism is dominant. Now, now this process you mostly carry out in the batch enrichment batch enrichment cultures. Next is you're trying to enrich the cultures using the solidified media. The solidified media techniques have been used for the isolation of certain enzyme producers, and these techniques usually involve the use of a selective medium incorporating the substrate of the enzyme, which which encourages the growth of the producing types. Thank you.